This video will cover how to use the spatial signal operation in Danse. Specifically, we will define spatial signal, then cover the prerequisites, demonstrate how to use the function, how to save and export figures, and finally how to automate the operation with Experiment Analyzer. Use the following timestamps to skip to the relevant sections. In Danse, the spatial signal operation is a heat map that combines animal coordinates with the signal of interest. In this heat map, the x and y axes are the width and length of the cage, while the color indicates the strength of the signal. The spatial signal is computed by averaging the signal at each time point when the animal is in a particular spatial location. This operation is quite useful for a number of analyses, such as condition place preference, the plus maze, novel object, animal interactions, hippocampal place cells, among many others. There are two prerequisites when using the spatial signal operation. The first is to have a continuous signal, such as raw neural data, delta f over f, animal speed, etc. The second is to have our animal coordinates, either the body, nose, tail, etc. These coordinates can come from several different sources, either calculated within Danse with the animal tracking operation that uses a threshold to detect the animal, either by importing animal coordinates from a CSV file from sources such as Ethovision, Animaze, externally trained Deep Lab Cut, or others. Or they can be generated with our animal pose estimation function, which is the Deep Lab Cut operation embedded in the software. Once you meet both prerequisites, select the Analyze button. This opens a window where we can select the type of analysis that we want to do, in this case, spatial signal. Next, we can specify the path to the continuous data within the file manager. Start with the data folder, the driver when necessary, the group, the series number, and finish with the dataset. Then we can select Analyze. In the second pop-up window, the tracking parameter specifies which coordinate will serve as input, especially if you ran the animal tracking operation multiple times with different settings or if you have multiple cameras. The body part parameter specifies which coordinate between nose, body, tail, if you have multiple, will be used to create the heat map. Here we only have body part. The field parameter specifies over which portion of the image the heat map will be created. Select video image to include the entire frame. Select the arena to only include the portion of the image in the predefined arena. Select custom to further adjust the field location by specifying the coordinate of the top left corner where 0, 0 corresponds to that first pixel, and the field size and width and height in pixel. The final parameter, field binning, allows you to define to what extent you will average nearby pixels together. The larger the binning value, the greater the neural signal will be averaged over a bigger area. To create our plots, select Apply. Here we can see the output plots. This plot is saved under the Data Analyzer folder as Body Spatial Signal. To export the figure, select the Save Plot icon. Or use the Save CSV icon to save the values behind the plot. Here we have a 10 by 10 grid where each value is the average delta f over f for each pixel. Note that the same operation is also available in Experiment Analyzer under the Create Pipeline tool to quickly apply the operation over many data files. We can also use the Group Analysis tool to pool spatial signal data from multiple animals together according to the experimental design. For more details on Experiment Analyzer, check out our Getting Started tutorial video. If you have any additional questions or concerns, please reach out to us at support@doriclenses.com.